I'd like to welcome you all to the Wednesday afternoon lecture series. My name is Laura Cayley. I am a senior investigator in and the branch chief of the Social and Behavioral Research Branch in the National Human Genome Research Institute. Today's lecture is special for at least two reasons. One reason is that this is an NIH Margaret Pittman lecture, which I will expand upon briefly in a moment. The other reason is that our speaker, Dr. Gigi Lozano, can finally join us in person. We nominated Dr. Lozano approximately 2 years ago now. She was scheduled to speak during last WALS season, but postponed that visit to be sure that she could deliver a WALS talk in person. And so here she is in the flesh. It's been really exciting over these past few weeks, planning for her visit, building up for this lecture, and we're really excited to have her here. Dr. Rotimi will introduce our speaker today, but I wanted to say a few words about this remarkable person, Margaret Pittman, for whom this lecture is in honor of. Margaret Pittman came to medicine early in her life, helping her father, who was a country doctor in rural Arkansas, and she became smitten with biology. After earning a bachelor's degree in biology and mathematics, she went into teaching, and she used the money that she saved from teaching to go to the University of Chicago and earned in 1928 her doctorate in bacteriology. Dr. Pittman came to the NIH in 1936 by way of the Rockefeller Institute and the New York State Department of Health. At the National Institutes of Health, her career path led her to become the very first woman to hold the position of laboratory chief, heading the Laboratory of Bacterial Products, Division of Biologics and Standards from 1957 to 1971. Dr. Pittman isolated the influenza strain responsible for most childhood meningitis, helped identify the cause of epidemic conjunctivitis, and made key observations that led to the development of a salmonella vaccine. In 1970, Margaret Pittman was recognized with the Federal Women's Award, and she served as the president of the Society of American Bacteriologists and of the Washington Academy of Sciences. Although Dr. Pittman retired in 1971, she kept working at the NIH as a guest until 1993. Thus, she contributed to the scientific community for almost 50 years. The Margaret Pittman Lectureship, created in 1994, honors Dr. Pittman for her exceptional research achievements at the National Institutes of Health. As well, the Margaret Pittman Lecture honors a researcher who exemplifies the intelligence, scientific excellence, and drive of Dr. Pittman, and who is dedicated to advancing and improving the careers of women scientists. Dr. Lozano is most certainly deserving of this honor. So on behalf of the Women Scientists Advisors, I would like to welcome Dr. Gigi Lozano to the NIH. We really are looking forward to hearing more about your work. With that, I'd like to introduce Dr. Charles Rotimi, who will introduce Dr. Gigi Lozano.
Thanks, Laura, for that wonderful commentary about Margaret Pittman. It is indeed my honor today to introduce our speaker, but before I do that, my name is Charles Rotimi. I'm the scientific director for the National Human Genome Research Institute. So before introducing our speaker, I have two quick announcements. One has to do with continuing medical education. The code is 44434, and I repeat, 44434. Okay, so please sign up and get your credit for attending this wonderful talk. Also, those of you who are watching remotely, via the NIH video cast, you can submit questions to the speaker. Simply click on the button that says Send Live Feed. which is below your videocast window. Type in your message, and we will relay it, to our speaker, at the end of our lecture today. Now, my tremendous honor, again, to introduce our guest, Dr. Lozano, is the Hubert L. Olive Stringer Distinguished Chair in Oncology in honor of Sue Gribble Stringer at MD Anderson. A renowned geneticist, she is recognized for her studies of the p53 tumor suppressor pathway, which is undermined in a large percentage of human cancer via mutations and deletions of p53. Using mouse model, Dr. Lozano and her team identified p53 proteins as a transcriptional activator. They showed that Mdm2 and Mdm4 proteins are potent inhibitor of p53. Over time, her lab studied multiple p53 mutations, extensively characterizing the protein's effect on the tumor suppression and tumorigenesis. Her groundbreaking work has provided invaluable testing novel therapeutic interventions. Dr. Lozano received her undergraduate degree in math and biology from the University of Texas, Rio Grande Valley, formerly the Pan American University. She received her PhD in biochemistry from Rutgers University and conducted postdoctoral training at Princeton University. I should note that Gigi, that's what we call her. Let's just say that she spent some time here at the NIH, and that is where we think she received the bug of, what I call the scientific bug and the research bug. And she was at NCI in Building 37. And I was just talking to Michael Gottesman, and he remembers all of the story about Gigi and the interaction with NCI. So hopefully maybe at some point we'll talk about that. So just to complete the introduction, Dr. Lozano has picked up many accolades and honors along the way. She's a fellow of the American Association of Cancer Research Academy and a member of the American Academy of Arts and Sciences, the National Academy of Sciences, and the National Academy of Medicine, and the recipient of the President's Leadership Award for Advancing Women and Minority Faculty at MD Cancer Center. In 2020, Cell named Dr. Lozano one of the most inspiring Hispanics and Latinx scientists in America. This year, Dr. Lozano received John Manderson Award for Faculty Leadership at MD Anderson. And I would be very remiss not to say that at a personal level, as the scientific director of NHGRI, Dr. Lozano serves on our Board of Scientific Counselor, and we have just tapped her services again to be on our Blue Ribbon panel. So the title of our talk today is Mutant p53 Activities in Mouse Tumor Models. Gigi, welcome back to the NIH.
Thank you, Charles, for that wonderful introduction. It's great to be back. I really, NIH was my very first lab experience. I learned molecular biology here, and I think I'm just very glad to be back and to see some old friends and to thank you for your contributions to my career. So today what I'm going to do is talk about some of our models, and I've changed the title just a little bit to include some wild-type p53 activities, because we're trying to really understand not just what wild-type p53 does, but what happens when it's not there and what other activities mutant p53 might have. So I think this will work. Okay, these are my disclosures. So I'm going to first introduce the p53 pathway. Hopefully, well, I wonder if I have an arrow. Probably not. Okay. So the p53 protein is present at very low levels in normal cells. It's hardly detectable. But stress, DNA damage, and inappropriate activation of an oncogene will unleash a series of kinases that phosphorylate p53 and make it a very stable protein. And then in turn, p53 functions as a transcription factor, a tetrameric transcription factor that activates hundreds of genes, and I only show some of them on this slide. And these genes have important cellular functions. Probably the first one that was characterized was the ability to initiate apoptosis. So p53 can kill the cell, can also kill the cell by another process called ferroptosis, but it can also arrest the cell cycle. So it can activate proteins such as a p21 cell cycle inhibitor and cause the cell to arrest or undergo senescence. And again, this cell cycle arrest is not a dead end, but senescence is a dead end. In addition, p53 has the ability to regulate its own levels, and so there is even a pathway in which p53 activation will lead to survival of the cell. And that is mostly through this protein called Mdm2. Mdm2 is a p53 target, and in turn, Mdm2 encodes an E3 ligase that targets p53 for degradation. So I think with this myopic view of the p53 pathway, I think you can begin to see that p53 has so many choices in a cell. It can kill the cell, it can stop the cell, and it can just say, time to go on. And even though we know some of the circumstances where p53 makes these decisions, most of the time we can't guess what p53 is going to do in the cell. So the first few slides, I'm going to talk about the physiological transcription program, because we were interested in trying to understand what the targets are, and could they have potential roles as tumor suppressors in and of themselves. It's been very hard to reintroduce p53 activity into tumors, and the idea was maybe we can reactivate an important downstream tumor suppressive pathway. So I have to also introduce the Mdm2 mouse, because this is a tool that we use in the lab. So deletion of Mdm2 is an embryo, whoops, wrong slide. Deletion of Mdm2 is an embryo lethal. The embryo, oops, I'm pressing the wrong button. I thought I was pressing the right button. Okay. Mdm2 loss leads to an embryo lethal phenotype at the blastocyst stage. This cell is undergoing apoptosis. Every one of those cells is dead. And we asked the question, what is the role of p53 in this process? And if we delete p53, we get a perfectly viable mouse. So this really cemented the relationship between Mdm2 and p53. And in fact, we now have conditional alleles, and we've taken Mdm2 out in any tissue. It is always a cell lethal. And it's always in a p53 dependent fashion. So what we've done then is we've used a model in which we use tamoxifen to express the Cre recombinase and generate an Mdm2 null in the whole mouse. And within just a few days of tamoxifen injection, all the animals are dead, shown here by the red line. And this is a p53 dependent process, because if you remove p53, the animals are perfectly viable. So this allows us then to ask, what happens, what is the p53 physiological response upon Mdm2 deletion? And we did this in a similar manner, but in an acute way, so that 24 hours after the last injection of tamoxifen, in which we have about 50% of the cells that have lost Mdm2, we can evaluate that transcriptional program. And the first thing we did was just look at the pathologies of several of the tissues in the mouse. And just 24 hours after recombining and losing Mdm2, you've got all these fascinating pathologies. The heart and the ovary, I'm pushing the right button. The heart and the ovary are perfectly normal. They don't show any phenotypes at this time point, but you can see the intestine, kidney, and the pancreas have all kinds of phenotypes. In the intestine, we see loss of the crypts. In the kidney, we see dilated tubules and protein casts, which suggests there's dysfunction there. And in the pancreas, we see this acinar to ductal metaplasia. So then we looked at the transcriptional targets of p53, and we identified hundreds of targets. So for example, in the pancreas, we identified 206 p53 targets. in the ovary, which had no phenotype, we identified 21 targets. And these are p53 direct targets because we basically took the RNA-seq data and combined it with ChIP-seq data that was available in the literature. And so this is the p53 transcriptional physiological program. So I think there are two important points I want to make. There's only 7 genes that are in common between these 5 tissues, and MDM2 is one of them. So it's very interesting that MDM2 is regulated by p53 in this physiological system in every tissue that we analyzed. What it's probably doing is balancing that p53 levels, maybe at different times during the cell cycle. Cyclin G was another one of those universal targets. POK, which is a polymerase. EDA2R is a cell surface receptor involved in cell death. But to me, the other fascinating aspect of this slide is how many targets are tissue-specific. So for example, again, in the pancreas, 135 transcriptional targets were only expressed in the pancreas. So that tells you that there's this huge diversity of transcriptional targets that p53 can regulate. We've done one additional validation experiment to ask if this is a true p53 response by irradiating the whole mouse and isolating these same tissues. And you can see here in the middle is a wild-type mouse that was radiated. So those are the samples that the transcriptional targets, okay, I'm not going to use the pointer anymore. So that middle portion is wild-type p53 after radiation, and those are the cyclin G and the EDA2R targets. They're upregulated in a wild-type irradiated mouse, but not in a p53 null mouse. So this is not just, this is the transcriptional program with loss of MDM2 and in the presence of radiation. So the other, I think, major finding was really when we tried to analyze this acinar to ductal metaplasia in the pancreas. So we're fascinated by this phenotype because within 24 hours, we see a complete sort of rewiring of the pancreas from normal at the top left to this ductal structure. And when we stain with a ductal-specific marker, CK19, you can see a lot of brown staining. So this is a plasticity, a transition that occurs. And the data are quantified on the right. So the question we wanted to ask, we wanted to develop a model where we were just looking at this transition in the pancreas. And so what we did is we used an acinar-specific Cre recombinase, the same model. This is a MST1 Cre ER transgene. So we add tamoxifen, and only in the acinar cells do you see deletion of MDM2. When we compare the recombination frequencies between the first mouse and this mouse, they're very similar. So we are deleting MDM2 and activating p53 in the pancreas, but we completely lost this acinar to ductal metaplasia. So now the tumor looks, you know, the normal mouse pancreas looks absolutely normal, and the immune component is also very normal. So to me, this is an important finding because it's the first time that we realize that there are communication, that the transcriptional program isn't the same for every cell in the pancreas, and that there are independent, non-cell autonomous effects that give rise to this plasticity. So it's one of the first times that we've been able to actually see communication between p53 transcriptional program in different cells. So to summarize these data, I'm fascinated that p53, I mean, it is a transcription factor. You expected to have hundreds of genes that were regulated, but I didn't expect to see so many regulated in a tissue-specific manner. It's also showing us that p53 transcription affects cells in a non-cell autonomous fashion. And so one of the things that we've just initiated is some single-cell analyses of the pancreas to determine what cells are expressing which transcription factor and how are they communicating with each other. But the major part of my talk today really is on mutant p53 activities and what is mutant p53, how is mutant p53 causing cancer. So the first few slides just basically tell you how important p53 is as tumor suppressor. This graph we call the Manhattan plot because it looks like there's a bunch of skyscrapers. It was developed by a colleague, Magali Olivier, Across the x-axis, there's 125 genes that are mutated in cancer, and across the y-axis, there's 36 different kinds of cancers. And I think what you can clearly see is that that row of skyscrapers on the right-hand side of the slide is where p53 is located. So it's mutated in most cancers. And when p53 isn't mutated, you often have dysregulation of the pathway by other mechanisms. The other fascinating aspect of the p53 tumor suppressor is that it's not usually lost. Missense mutations are the most common type of p53 alteration. So this is a line drawing of p53, and these are the kinds of mutations that occur in breast cancer. It's similar for other cancers, but I'll be talking about breast models, and so that's why I'm showing this slide. The little black circles are truncating mutations, and then the green circles are all the missense mutation. And when I did the calculations, this is about 60% missense mutations. There are several hotspots. The 175, the 248, and the 273 amino acids are very, very often mutated in cancers. The arginine 175 is important for the structure of the DNA binding domain. Most of the missense mutations occur in that red domain, which is the DNA binding domain. And the 248 and the 273 are arginines that directly bind DNA. So altering those arginines disrupt the ability of that protein to bind DNA. The transcriptional activation domain is at the amino terminus of the protein, and it's intact in all of these missense mutations. And then the carboxy terminus contains the tetramerization domain, because p53 functions as a tetramer. And in that domain, you also see some missense mutations that disrupt tetramerization. So there are several features of these p53 missense mutations that I want you to keep in mind as I discuss our data today. Loss of function. These are true loss of function alleles. They do not express a protein that can bind DNA and activate transcription. They also have some dominant negative and inhibitory effects on tissues. So because in most of these DNA binding mutants, the tetramerization domain is intact, what you have is a mixed tetramers that have wild type and mutant proteins. And so that mutant protein basically inhibits the activity of that wild type protein. And I'll show you a very clear example of that. But then the other feature that's often discussed in the literature, and I think it's real, I was able to convince Glenn today that it was real, is what we call a gain of function. Basically what this means is that the mutant p53s have additional activities that contribute to that tumor growth. And oftentimes these activities are due to interactions with other DNA binding proteins. And basically you unleash a transcriptional program that's not a p53 dependent transcriptional program. So the first few slides are germline models with these p53 missense mutations, because we really, the R172H is equivalent to the 175, and that's that contact mutant. And then we have, that's the structural mutant. And then we have the 245 and the 270 mutants, which are the contact mutants. And we really wanted to ask a very simple question: are all mutant p53s the same? So this is a model in which we compared the 3 different alleles in the same background, C57 black 6, and we asked, they all get tumors, but do they get the same kind? So the first experiment that we did is actually look to see whether that wild-type protein activity varied depending on the presence of that mutant p53. So this is an experiment where in vivo we've taken a whole mouse with the different genotypes, we've radiated the mouse and looked at the ability of that p53 to activate transcription. So the first gray bar is the irradiated tissue, and this is probably the thymus, and that shows that, for example, here in the backs, p53, the backs p53 target is activated 8-fold above background. And then you see the controls, which is the white bar, is the heterozygous mouse, and then the 3 different mutants were analyzed subsequently to that. And I think as you look across the entire slide, you can see that those bars with the mutant p53 and a wild type allele are always lower than the heterozygous white bar. And what that says is that in response to DNA damage, that mutant p53 is actually dampening the effects of that wild-type p53. So this can contribute to an increased tumor phenotype because you've got less wild-type p53. And in fact, when we look at the survival of these animals get tumors. I'll tell you which kind in a minute. But you can clearly see on the left, when we look at a little over a year, the samples that have the blue and the black, the 245 and the 270, these are the contact mutants. They get cancers earlier than the heterozygous mouse and the 172 mutation. So there is a difference in terms of the tumor onsets in vivo. And in addition, the kinds of tumors that they get are all very similar. They get lymphomas, sarcomas, and they get some carcinomas. Remember, we're in the same genetic background. But the other major phenotype that we see with the mutants, all three mutants, is a metastatic phenotype. And Tyler Jacks's lab and mine described this phenotype as the mutant p53 gain-of-function phenotype years ago. So the different mutants, I think I summarized. Okay, this is one more slide from these data is one of the other concepts that I want you to take home from this talk is that mutant protein has to be present at high levels in order for it to dampen that wild-type p53 and in order for it to contribute to this gain-of-function metastatic phenotype. So what we've done in this experiment on the right is we've compared animals that are homozygous for these different alleles, and we bin them into whether they expressed less than 40% of stable mutant p53 on the left or very high levels of p53 on the right. You can see a tissue stained with p53 by immunohistochemistry, and 91% of those cells have very stable mutant p53. And when we bin these into these two categories, less than 40% stable p53 in cells or greater than 60% in cells, there's a very big difference in survival as well. And the samples that have very stable p53 in red, those mice are getting cancer sooner than the other animals that have less stable p53. So this is one of numerous pieces of data that suggests the stability is contributing to the lethal effects in these cells. So just to summarize this bit of the talk, we've made germline mutations. They mimic the Li-Fraumeni syndrome. And all three of those mutant proteins, when stressed, dampen the activity of wild-type p53. The 245 and the 270 mutants are more aggressive. They have worse outcomes than the 172 or the p53 null allele. And that stability of that mutant p53 leads to the decreased survival of those animals. So we've studied the germline almost ad nauseum, looking at different p53 missense mutations and other alleles. But what we really wanted to study is a somatic model, because somatic cancer happens more often than germline. And what we really wanted to do was to develop an animal model on the right where you can make a mutant p53 only in a few cells surrounded by a wild-type compartment. And for us, it was interesting. We really wanted this mouse to be able to understand the tumor microenvironment interactions. And so in the animal that I will show you in a few minutes, the neutrophils, the CAFs, the immune system, the T cells, are all wild type for p53. So this is how we generated those animals. We call it the p53WM allele because it converts wild type p53 to mutant p53. And the way we generated these alleles is to basically upstream of the point mutation, which is that asterisk in exon 5, we inserted a wild-type cDNA followed by a polymorylation site. And so normally this allele would express a wild-type p53 protein, and then in a pre-mediated fashion, you would remove those wild-type sequences and express the mutant protein. And we did this both for the structural mutant, the 172, and the contact mutant, the 245, which is really the most aggressive allele that we have in the lab. So this is just one of many slides that I won't show you that basically shows that the animals that inherit one of these WM alleles, either the 172 in blue or the 245 in red, have no differences to a normal mouse. So these are not tumor-prone models, which suggests that you're really making a wild-type p53 in these mice. Okay, so the first experiment I'll describe is one in which we've done a breast model, and what we've done is we've injected an adenovirus expressing Cre into the duct of the mammary gland. And on the left, if we use low titer, we can get Cre recombination of TD tomato in a few cells, and then on the right, if we use a higher titer, we generate a whole bunch of cells that are positive for Cre and TD tomato and mutant p53 when we put mutant p53 in the system. Now, the other point I want to make with this slide is we inject always females when they're 10 to 12 weeks old, and we use a background that is 50% C57 and 50% BALB-C. And the reason we use a BALB-C background is because the BALB-C mice are susceptible to breast cancer, whereas the C57 strain that we've been working with all along rarely gets breast cancer. So there's something about this genetic background that synergizes with the p53 mutation to yield breast cancer. So these are the data. Again, these studies showed that there were differences between the alleles. So the 172 mice on the left, for example, heterozygous mice, because we wanted to mimic the somatic mutant p53 without LOH, we never saw any tumors. If we irradiated that mice and generated random mutations, we begin to see tumors. But look at the right-hand side of that graph. The 245 heterozygous all by itself at low dose and at high dose yielded breast cancers. All of the mice that had mutant p53 had metastases, but again, the 245 was the most metastatic lesion. And the other fascinating aspect of the story is on the right there. So you don't always see LOH with the 245, but you always saw LOH with the 172. So to me, this suggests that there are differences in the ability of these mutant proteins to dampen wild-type p53, and sometimes you need to lose that allele, and sometimes you don't. So you need to dampen the p53 pathway, and you probably do it by multiple mechanisms. The other, I think, fascinating phenotype that we're struggling with and trying to understand is that right column here is, so the 245 mutant gave us 3 different kinds of breast cancer. Black bar is triple negative breast, and then in dark blue is HER2 enriched, and then in light blue is likely luminal B. So to me, this is fascinating because we start out with the same p53 mutation in all of these cells, and yet tumors evolved in 3 different directions. And I don't need to tell this audience that triple negative breast cancer is hard to treat, but HER2 and luminal B, there are hormone therapies to treat those tumors. So we've embarked on a big genome study to try to understand the RNA and the DNA transcriptomes to see what are the changes that are driving these 3 different kinds of breast cancers. So to conclude this part of the study, mutant p53 models develop metastatic breast cancer. I mean, this is just a beautiful model of what happens in humans. The 245, as I just indicated, leads to the development of 3 different kinds of breast cancers. And the data I didn't have time to show you, because I do want to get to the other part of my talk, is that the metastases in this model are seeded very early. The initial study included, I think, about 20 mice, and we've now expanded that study to about 100 animals with the 245 mutation. And we've sacrificed most of these mice. And some of them we've sacrificed because they had difficulty breathing, and the veterinarian said we had to sacrifice the mouse. When we open up those mice, we have a lot of lesions in the lung, but we can't find a primary breast tumor. So what we think that this is just another example of early dissemination of that primary cancer. So the last model I'll discuss with you today is one in which we've used this system to basically turn on that mutant p53 and then take it away. And we want to ask the questions, are those tumors dependent on having that mutant p53? So we start out with the wild-type p53. These are these WM alleles. We then, in this model, we use K14 Cre, so we're expressing Cre recombinase in the entire epithelium of the breast and the the salivary glands, and the skin. And then a year later, we get breast cancer. We also get some head and neck cancers, but we haven't studied those. And then for the breast models, because we've also turned on Cas9 when we turned on K14 Cre. So now we're able to come in with a guide RNA, the adeno-associated virus expressing a guide RNA to take out that mutant p53, and we can ask, what happens to the cell when you remove that mutant p53? So importantly, these mice only express one mutant p53 and one null allele. So there's no wild type p53, there's no allo-H. We did it from the beginning. Mutant p53 is only expressed in this system. And some of the mice also have TD tomatoes, so we can monitor the primary tumor development and the metastases. So the K14 Cre model gives us triple negative breast. All the mice are dead within like a year and a half. They get two kinds of breast carcinomas. One is this adenocarcinoma, and the other is in a sarcomatoid-like tumor. And they have very different etiologies. So what we've done then is injected these tumors after they developed with the adeno-associated virus expressing a guide RNA to the mutant p53. So we're depleting the tumors of mutant p53. And on the left, I've got a graph that shows the controls in A. So this is what happens when mutant p53 is still being expressed. And you can see that the tumor volume continues to increase. In purple are the tumors that now have depleted mutant p53. And I think you can see a big difference between those bars. So some of the tumors have a decreased volume, and some have a large decrease in how quickly that tumor is growing. On the right, you see the tumor volume with time. Again, in purple are the mice that have depleted mutant p53, and in green are the mice that have high levels of that mutant protein. In orange are two interesting samples that look like they still have mutant p53. They're continuing to grow. These were two samples for which the mutant p53 was not stable. And so you need to have a stable mutant p53 for the tumors to be dependent on that mutant p53. Next is just a survival curve. So in purple are the animals for which we removed that p53 protein, and you can see that they're living a lot longer. This is probably an underestimate of how long they live, because some of these animals get secondary tumors, and we have to sacrifice them because of the secondary tumor and not the primary tumor that we treated. The other mutation, the 245, it's the same story. You delete that mutant p53, and you can see in purple that these tumors either regress or drastically slow down. And you can see on the right in the survival curve, you also extend survival of these mice when you remove that mutant p53 protein. And this is the experiment when I want to show you the responders versus the non-responders. So you have, this is a section through the tumors. The AAV control is on the left, and everything in red is p53. So the two responders in the middle, you can see that huge hole that doesn't have any staining, and that's where we've depleted the mutant p53. And both of those tumors regressed, the volumes regressed. And on the right, you have a non-responder, but you can clearly see that there's no stable mutant p53 in that non-responder. So this responder looks like it had no p53 to start with, so it wasn't dependent on that mutant p53. So I think that this is an important experiment, because I think in the clinic we need to think about not just the point mutation, but whether it's become a stable protein or not. So we might have to revert back to immunohistochemistry to detect mutant p53 stability. So the mutant p53 proteins, both the 172 and the 245, are required for maintenance of these tumor phenotypes, because when we take these two proteins out, tumors regress, and most of these tumors have stabilized that mutant p53, a few do not. And I think that's another important question in the field that we've not been able to fully understand. What is it in these tumors that is causing the stability of that mutant p53? Is there some stress, some damage, is there inflammation? Are those contributing to the stability of that mutant p53 protein? And then if the tumors have an unstable p53, they're not dependent on that mutant p53 for continued growth. So now that we know that taking out mutant p53 causes these tumors to regress, we're really interested in trying to understand how that happens. So in this experiment, what we've done is we've, in purple again, we've treated these tumors with the AAV-associated guide RNA that depletes mutant p53, and you can see that the tumors don't grow as quickly. This is just 6 days, so it's very early on, because we wanted to catch the early events to try to understand what the trigger was. So in the middle panels, what we just did some H&E, and we looked at these as these tumors, and when we deplete mutant p53, we can see these very large lipid-like structures, and they're quantified on the right. So when we deplete mutant p53, we see these large structures, very reminiscent of a process called ferroptosis, which is a cell death mechanism. So the other experiment that we did is we stained these tumors with 4-H&E, which is basically a marker for lipid peroxidation, and I think you can clearly see all that brown staining in the middle. When we deplete mutant p53, we have a very high level of lipid peroxidation. So these bits of data are suggesting to us that maybe we're dealing with a ferroptotic cell death mechanism. So the next experiment was simply to isolate those tumor cells away from all the rest of the microenvironment to see what was actually happening in those cells. So we did some single cell analyses. We've identified 3 clusters, 1, 11, and 14, that were the tumor cells. And on the right, you can clearly see that in the control, these clusters are present at high levels, and when we deplete mutant p53, these 3 clusters are lost. So these are the clusters that are the tumor cells that express the mutant p53 protein. So of course, you do all these analyses, and you have to look at the transcriptome. And in these samples, with depletion of mutant p53, we saw upregulation of reactive oxygen species. And so again, this is a mechanism that increased ROS leads to a ferroptotic response. So we're beginning to think that ferroptosis is the mechanism of cell death. So what is ferroptosis? It's a very complex cell death mechanism. We're still learning a lot about ferroptosis. It's basically too much reactive oxygen species causes this lipid peroxidation. It's an iron-dependent process, and basically it kills the cell. So on the left, you can see a lipid molecule with a lot of hydroxyl radicals, and that will lead to ferroptosis. There's an important enzyme called GPX4, which is a glutathione transferase, which basically removes those hydroxyl radicals and allows the cell to survive. So it's a very critical protein in the cell that maintains the peroxidation at a level that's tolerated. So the next experiment I'm going to show you is one in which we've inhibited GPX4. So basically, we've induced a ferroptosis in these cells. So what we've done here, it's an interesting experiment, because we have in green cells that either express the mutant 172 or the mutant 245, and then we used CRISPR to remove mutant p53 from these cells. And my postdoc came to me and said, I have cells with deletion of mutant p53. And I said, but in vivo, those cells are dying. How did you get those cells to live? And it turns out they're very fragile. We have to change the media every day because it turns very yellow. And so they've tolerated loss of p53 in culture, but they're very fragile. And then when we add RSL3, this inducer of ferroptosis, you can see that the cells that have no p53 deletion, are very sensitive to ferroptosis. And I think the dramatic sample on the far right is one in which the cell that has the mutant p53 is resistant, and the cell that has lost p53 is it dies very quickly. So we've also inhibited ferroptosis in vivo. Okay, so this is an experiment that I've previously shown where you can see if you deplete mutant p53 in purple, these animals live longer. So what we've done is we've depleted mutant p53, and then we added lipostatin, which is an inhibitor of ferroptosis. And you can see those orange lines moving closer to the green line. The data are quantified in the middle. So when we prevent ferroptosis from happening, those animals are dying sooner. And that data then is shown on the right. The mutant p53 depleted tumors continue to, they live longer. The mice with mutant p53 depleted live longer, but if you induce ferroptosis, they're dying. So I think this is very clear. And each one of these is it's not a syngeneic mouse. It's a real mouse that developed a tumor, and then once we saw the tumor, we treated it with the lipostatin. So the hypothesis that we're working with is that mutant p53 mitigates this oxidative stress, and it protects the cells from ferroptosis. We remove that mutant p53, and the cells, the tissue, the cells are dying by ferroptosis. So how to test this? So the first thing we did is we went back to our single-cell RNA-seq data, and we looked at the reactive oxygen species, the genes that are responsible for ROS. And there are two genes that stuck out, PRDX6 and MGST3. And PRDX6 is a glutathione-dependent peroxidase, so again, it's going to work similarly to GPX4 and remove those hydroxyl radicals. And MGST3 is a microsomal glutathione S-transferase gene. So both of these have functions similar to GPX4 in that they're removing the oxygen radicals from the system. On the lower left in this slide, what we've done is we've compared the mutant p53 expressing cells and those with mutant p53 depletion and basically validated the data from RNA-seq. So deletion of mutant p53 causes downregulation of these two important glutathione enzymes. So in these cells, now in syngeneic mice, we've knocked down MGST3 or PRDX6, and the knockdown data are on the left, so we get pretty good knockdown. And then on the right, we've monitored tumor growth. So you can clearly see in the red and the green bars that if we knock down these two enzymes, we've sensitized the cells to ferroptosis. Okay, so what we think mutant p53 is doing is basically by allowing the expression of these two genes, mutant p53 is maintaining a state where the peroxidation of the lipid radicals is tolerated by the cell. So if you remember that gain-of-function slide, one of the ways that mutant p53 can contribute to gain-of-function is basically by interacting with other transcription factors. So we took our RNA-seq data and we asked, there's different databases that you can use to basically ask, is there any similarity between all of these transcriptomes? And so what we identified in this case is four different promoter sequences that were present in these genes that are upregulated upon depletion of mutant p53. And the one that caught our attention in this case is this NRF2 protein. And NRF2 is a DNA binding protein, and about a third of the genes that were differentially expressed upon depletion of mutant p53 had an NRF2 motif. And NRF2 is in the ferroptosis pathway, and I'll show you where it fits in a minute. So the next question we asked, because this is the hypothesis that we're working with, is that mutant p53 interacts with other transcription factors. to drive a specific program. And so these are two different assays in which we look to see if mutant p53 could interact with NRF2. So on the left is immunoprecipitation experiments in cells that express the 172 mutant protein. And you can see that if we immunoprecipitate with p53 in the rightmost lane, we can detect NRF2. And then on the right was some in vitro data where basically we expressed these proteins in vitro, purified them, and again, in the rightmost panel, you can see that a his-tagged NRF2 protein is interacting with the 245W tag. So and we've done this for both the 245 and the 172 mutants, so it doesn't matter which mutant you're expressing in the system, that mutant protein is interacting with NRF2. So this is it's not a chip assay, it's this newfangled cut and run assay where again, you're asking the same question, is the mutant protein, mutant p53 and NRF2 present on the same DNA sequences? So we took 2 of these ARE elements from the PRDX6 gene, site 1 and site 2, and you can see that NRF2 on the left is bound to the sequences, and on the right you can see that when mutant p53 is present, it's also bound to this sequence. And so this was, I think, very convincing data, although we're having a hard time convincing the reviewers that mutant p53 is interacting with NRF2. So here's what we think is happening in the cell. So in the lower left-hand corner, you have mutant p53 interacting with NRF2, and it's driving expression of these 2 proteins, MGST3 and PRDX6. And these proteins in the middle green function similar to GPX4. They're responsible for removing some of these hydroxy radicals on lipids and allowing the cells to survive. So what happens if you transition to the right side of this slide is you remove that mutant p53 and you no longer have expression of these 2 proteins. You can see from the single cell data and the transcriptome data that these 2 proteins were down modulated upon depletion of mutant p53. So what we think is happening there is you've offset that balance. Now you only have GPX4 trying to deal with this increased reactive oxygen species and hydroxy radicals, and it's just not sufficient. And so removing that mutant p53 is taking cells down that pathway. Now, one additional side story that I won't give you a whole lot of details, but I think it's, again, fascinating with respect to the complexity of the system. So I told you at the beginning that these mice get these adenocarcinomas and they also get these sarcoma-like tumors. So in both instances, when we remove that mutant p53, we get depletion of the tumor volume, and the animals live longer. But for the adenocarcinomas, I've shown you all the data for the adenocarcinomas. It seems to be ferroptosis. But if we look at the sarcoma data, it's not ferroptosis. And so even though those tumors, which are driven by the same p53 mutation, they develop differently. They give rise to a different pathology. And while they're dependent on p53, they're not dependent because of a ferroptosis. So that just gives you one more example of the complexity of working with p53. I think there's going to be a lot of tissue specificity. So hopefully I've given you a smattering of some of the studies that we've done. I'm fascinated by wild-type p53 activities. What is the transcriptional program that p53 can initiate in different cell types? We're hoping that that might lead to the ability to activate that pathway downstream and potentially contribute to tumor suppression in some way. And then I've talked about a lot of the different p53 missense mutations, some of them in the germline, and they're different. They're not the same. And I think the other fascinating aspect of all of these models that were generated is we never know what tumor type we're going to get. So I think trying to understand the evolution of those tumors, trying to understand some of the contributing lesions. And so some of the studies ongoing in the lab are trying to understand those early lesions and what are the changes that happen early on that contribute to disease, Like I said, we're sequencing a bunch of these tumors to try to understand the genome as it evolves. And then lastly, there was one more. We really want to understand the metastatic process, because that's ultimately, I think, what kills a patient besides drug resistance, Michael. And so we're sequencing some of those mets in the lung and in the liver to see what changes. It's not an easy problem. I think metastasis is very difficult to study. I think we've studied it in tissue culture way too long, and I think we need to study it in vivo systems where we're watching the process. But again, we have to mark these cells with TB tomato, and we have to sacrifice a lot of mice early in the process to try to understand that evolutionary process. So with that, I'm going to just, I think, thank the people in my lab. This is a new picture. Ever since just a few weeks ago, we dropped the mask mandate at MD Anderson, and we quick took a picture. So Donata Deeper did all the studies with the ferroptosis, and it was really her work looking at the sequences, looking at the samples that suggested that ferroptosis was a mechanism by which mutant p53 was maintaining these tumors. Peirong is a long-term technician in my lab. She's absolutely wonderful. Shunbin did the genomic studies that I described. Sydney, who's finished her PhD and moved on, so wasn't here for the picture, did the transcriptome studies. And I work with two incredible pathologists, Adel and James, who never hesitate to look at the tumors that we bring them. So with that, I'll stop here and I'll take questions.
Thank you. Anyone's certainly free to use the mic. Let me start off with a question from online, if you don't mind. It's from Han Mo. And can you comment about isochromosome 17q in myeloid neoplasms? These tumors always lose one copy of TP53, deleted, but we never found mutations in the second copy. Mm-hmm. The behaviors of these leukemia are as bad as the TP53 biallelic lost ones.
Yeah. So I know that's true. None of our models get leukemia. So I think that, especially the germline models, I think that the other cell types are more susceptible to p53 mutations in the backgrounds that we're working with. It's a fascinating problem, and I don't know the answer. It's doing something. And those are the worst leukemias when they have a mutant p53. So again, I think context is very important, and I think we need to look at the transcriptome in the context of that mutant p53 and how it's changing. But I've not worked on AML, but I do want to. We'll get there.
Yeah, so it's beautiful. And I'm an immunologist, so I will have an immunology-related question. Have you looked in this tumor model into a RAG deficiency, or how much is the immune system implicated?
Okay.
So one thing is a very long timeline. Mm-hmm. So have you also looked at how many mutations you accumulate in these models, and how many can induce a specific immune response?
Okay, so what we've done is we're just in the process of sequencing, so we're getting all that data. But the single cell data suggested that there was an immune response when we removed that mutant p53. Yeah. So we're trying to figure that out, but we haven't done any experiments to actually say how that immune system is contributing to the decrease in the tumor volume and the increased survival.
So, in related to that, do you see in how many cells is there a tumor mass which becomes critical in terms of whether there is a rejection or there is reduction of the tumor growth where you induce. So if the immune response is involved, do you think that how many cells will then be mutated is relevant?
Yeah, that's a great question. And one of the reasons that we developed the somatic model with the adenoductal injection of Cree is just to make a few cells become a tumor cell. And so I don't know the answer to the question, but I think we can use the model to try to address those questions over time, which is, I think, what you're asking. Yeah. Glenn.
It's a great talk. So I have a related question, or maybe a deeper question. So, you've done a fantastic job of looking at intrinsic or cell autonomous models, and mechanisms. But, you could almost look at every part of your talk and ask a question about the tumor microenvironment. So for example, do you know if the different mutants have different effects on either the immune system or other microenvironmental components? Does that play a role? What about at the metastatic sites? And of course, when you turn it on and off. But there's probably differences, and
Yes.
because they'll change secretions and all kinds of stuff.
Right, we're sequencing all of these samples. Invite me back next year. No, give me a couple of years.
Gigi, thanks so much for that lovely talk. Speaking about drug resistance, one prediction is that for your mutant p53 that has some way of blocking ferroptosis, you'd expect that the tumors would be more resistant to the normal stimulants, Erastin, for example, ferroptosis. Are they resistant to ferroptosis stimulators?
So, so we haven't done the RAS experiment in vivo. We've only done it in the tissue culture cells. At least I don't think so.
And in tissue culture, are they resistant?
But that's an experiment that we need to do. Yeah.
And we had a little conversation before about the fact that there are some mutant p53s that stimulate transcription of the PGP gene,
Yes.
my favorite
Yes.
gene.
Yes.
Do you know whether the one you're looking at is a stimulator? And if so, then the cells should be more generally resistant to
Right.
anti-cancer
Right.
drugs.
We have all of these sequencing data that we need to look at for exactly those kinds of questions. Yeah, yeah, yeah. Hi,
And I concur with everybody else. Wonderful talk.
thank you.
My question is, what happens in the context of a tumor virus where you have wild-type p53 and then a mutant that's unable to engage the viral protein like T antigen or E6,
Oh,
what
that's
have
an
you?
interesting question. Yes, so T antigen tumor model was generated years ago, and it was the first, in fact, the first cancer model that was generated. And we know that T antigen sequesters wild-type p53, prevents it from function, okay? And I don't remember exactly where in p53 T antigen binds, but I'm going to assume it's at the amino or the carboxy terminus, because that's where most of the interactions occur. You have the DNA binding domain bound to DNA. And so I think what would happen, and I don't know if anybody's done the experiment, is that the interactions of that T antigen with mutant p53 would stabilize that mutant protein. And then you would get, it wouldn't just be loss of p53, wild-type p53 activities, you would have a more stable protein that would contribute to a more aggressive phenotype. It was the prediction.
Right. And that would be the case for every, adeno, HPV, what have you, because I noticed in your
Yeah.
125 genes that are modulated,
Right,
cervical cancer and Burkitt's, which are viral tumors.
Right.
So,
right, yes. So there are, yeah. In fact, we published a really nice review, I think, just a few years back about all the mechanisms that can inactivate p53. And that's something that we need to think about because, HPV, E6, I think, degrades wild-type p53. So to treat those cancers, you need to remove HPV so that the p53 can come in and do its work. Other mechanisms include overexpression of the Mdm2 or Mdm4, which are potent p53 inhibitors, and you see those in a lot of sarcomas. So again, there are Mdm2 inhibitors, and Mdm4 are being developed in the clinic to disrupt that Mdm2 p53 interaction. They've been a little bit toxic, and the toxicities were predicted from our models. Bone marrow, it's a highly proliferative tissue. You disrupt that Mdm2 p53 interaction, and you get depletion of bone marrow. And then, so it's the viral Mdm2 overexpression of p53 mutations. So there are several mechanisms, and I think that we need to be cognizant of how p53 was disrupted in order for to be thinking about how we're going to treat the patients. We've done a few studies in animal models where we've reintroduced wild-type p53 to ask, will it combat the development of those tumors. And what we see is that if there's no p53 to start with, so you have a deletion of p53, it's very good at suppressing that growth. But if you have mutant p53, it can't suppress. Or if you have, yes, or if you have high levels of MDM2, you can't get enough wild-type p53 in there to suppress growth. So I think those are all things that we need to consider when we move toward, clinical treatments. Yeah. Mhm. Yes.
I noticed that, you really present the story in some ways of p53, and particularly the mutant, as a redox modulator in the cell. But I wonder if the other side of the equation, the iron and the handling of iron in the cell, the transport of iron into the cell or out of transferrin, does that change in any way with these mutants?
That's a great question. We haven't measured iron in the system. I don't know how to do it, but I think we could find out. Yeah, that's a good question, because iron is required. And so,
Right.
maybe a different way of approaching the problem is.
Right, it's
let's
part of
remove-
that
Yeah.
Fenton
Yeah,
equation that,
yeah. No, I hadn't thought of that. Thank you.
right, thanks.
We'll go back and
Yeah.
think about experiments to do.
One more online, if I.
Oh, one more online?
Yeah.
Okay.
This is from my colleague, Roland Owens, who I think deliberately tries to make me tongue-tied, so here's my best I can do here. Glutathione can recharge ribonucleotide reductase. Have you looked at DNTP pools in any of these p53 mutants?
Okay, that's a great question, because there's some old data in the literature that wild-type p53 activates that reductase and causes changes in the DNTPs. But we don't have wild-type in our system. We haven't looked. We need to look. Yes, Sean?
I'm not saying that what you're presenting wasn't interesting. I loved it. I'm starting to get curious about those non-p53 tumors, and if there's if you've seen, or if there's a possibility that you initiate the tumor with these mutant forms, but then you can get loss of that and compensatory losses in say,
Ferroptosis.
ferroptosis. And so do you actually see in non-p53 dependent tumors that there's mutations in this ferroptosis pathway?
We didn't sequence those tumors, but I think that's a good point, because you should always sequence the opposite, the ones that aren't. No, we haven't done that.
You just might see
No.
pathway compensations.
Makes perfect sense. You don't just sequence the ones that give you the result you want, you sequence the ones that don't, to glean a little bit more from them. Yep. Okay, we'll go back home and sequence those 3 additional
Didn't
tumors.
mean to cost you more money.
Thank you.
Gigi, thank
Yeah,
you.
Rena?
Reflective of my ignorance of the
No,
field
no, no.
of p53
All
biology,
questions are
is there
good.
any role in stem cell homeostasis in each stem cells? Does p53 play any role in? And I'm thinking of those tumors that are hypothesized to be derived from stem cell aberrations.
So the p53 null mouse, for the most part, is viable. So, and it looks normal, and it's the right size, and it's tumor prone, but it looks developmentally normal. So in that scenario, I would say no. But there's an interesting phenotype in the liver, okay? And that is, if you delete MDM2 in the liver, and I told you it was a cell lethal, so it was the albumin promoter, so you delete it in those cells. And what happens in that scenario is that the remaining cells regenerate a liver. So they get rid of the cells that have too high levels of p53, but that sends a signal to the rest of the liver to regenerate. And so I do think that there's a wild type role in regeneration. I think that, but it's not at the cell level like you were implying. It's at the organismal level. We had some similar data ages ago in the intestine, because we take out MDM2 in the intestine, and in the young mouse, we punch all these holes in the intestine. They're very sickly. A week later, they're fine. And when we look at the mouse, every cell that had deleted MDM2 was gone, and we had this crypt function that increased, and so there is a compensatory mechanism to regenerate a tissue when p53 depletes the tissue.
Thank you.
It's a fascinating phenotype.
So
be
please
interested.
join me in congratulating our speakers and thanking them. Wonderful talk.
Thank you all. Enjoyed being here in person.